Very good morning to all of you. I am Krishna Priya, Assistant Professor, Department of Biotechnology, Dhanalakshmi Srinivasan College, Perambalur. Today we are going to see about plasmid vector. First of all, let's see what is meant by vector. Vector is nothing but a carrier molecule in which we are going to insert our gene of interest. So, next one is what is meant by plasmid. Plasmids are nothing but covalently closed circular molecule or extra chromosomal DNA. Other than the chromosomal DNA, there will be a DNA which is called plasmid. It is circular in nature, double stranded and self-replicating. First of all, we should know why, what is the need for choosing plasmid as a vector. First one is it is small, easy to handle, where it is very easy to insert our gene of interest into that plasmid. That is why we are choosing plasmid. Next one is easy purification. We can after transformation process, we can easily purify this plasmid vector from the host cell. Next one is straightforward selection strategies. Selection strategies are easy when compared to other vectors. Then next one is we can clone even a very small fragments less than 10 kb by using this plasmid vector. These are all the advantages of plasmid for using cloning vector. There is only one disadvantage, that is if you need to insert large gene of interest, means we can't choose this plasmid vector. We can choose other cosmid, phagmid and all. If we want to insert small gene of interest, means we can choose plasmid as a cloning vector. Next one is features of plasmid vector. Why? What are the important features which is present inside the plasmid? For that purpose, we are using that as a vector. First one is it contains an origin of replication. It contains origin of replication means which allows a replication of gene of interest of independently other than the host's genome. Next one is it contains selectable markers. There are two types of selectable markers, that is twin antibiotic resistant marker and other one is based on the colors we can differentiate which one is transformed and which one is not transformed, that is blue white screening method. And it contains a multiple cloning site. So, multiple cloning site means we can insert more than one gene of interest. Next one is it can be easily, as I was already said in that advantages, that is it can be easily isolated from the host cell. Next one is plasmid ranges in from 1.1 kb to 250 kb, example puck vector and tall vector and all. PBR322. It was one of the first vector to be developed in 1977. P stands for plasmid and BR stands for Bolivar and Rodriguez. They are nothing but the scientists who have discovered this PBR322. It is an artificial plasmid. 322 is nothing but to distinguish from other plasmid like PBR 322, 27 and 28. Size of PBR 322 is 4363 base pairs. It carries two antibiotic resistant marker which is used for selection purpose. One is ampicillin resistance, another one is tetracycline. In that selectable markers, there will be multiple cloning site. If we insert our gene of interest in that multiple cloning site which is present in the selectable marker, a process occurs called insertional inactivation. If we insert our gene of interest, the already containing ampicillin resistant power will be turned to ampicillin sensitive power. Based on this only, we can select which cell is transformed and which cell is not transformed. And it has a high copy number. It can be increased to 1000 to 3000. The vector comprising DNA, it can be derived from naturally occurring plasmids like ampicillin resistant gene is derived from R1 plasmid, and tetracycline resistant gene is derived from R6 5 plasmid, and the origin of replication gene is derived from PMB1 plasmid. This is the diagram. PBR322, which is the 4361 base pairs, and it has ampicillin resistant gene and tetracycline resistant gene. That ampicillin resistant gene contains restriction site for SEA1, protease vulgaris, PST1, SAL1, ECOR1. In this tetracycline resistant, BAMH1, PST1, like that. These are all the cloning site for insertion of our gene of interest with the help of the restriction enzyme. Next one. Okay. Screening by insertional inactivation of a resistant gene. This is nothing but a PBR322. There are two resistant selectable markers. One is ampicillin resistant and another one is tetracycline resistant and origin of replication. This orange color BB, this is the gene of interest which we are going to insert that gene into the plasmid vector. Okay, let us imagine that we are going to insert that particular gene of interest into the tetracycline resistant gene. Okay, ampicillin, first one is ampicillin resistant. Second one is also ampicillin resistant. First one is tetracycline sensitive. Why I am saying here tetracycline sensitive means we have inserted our gene of interest into the tetracycline resistant genes. I have already said that there is a process called insertional inactivation. If we insert our gene of interest into a particular resistant marker, that character of that, natural character of that particular marker will be gone. Now we have insert our gene of interest into tetracycline resistant. So after the insertion inactivation process, the tetracycline resistant power will be gone for PBR322. Based on this only, we will select our gene of interest containing cells, that is transformed cells from the non-transformed cells. Next one. Okay, this is a plate containing both transformed, non-transformed and all. Selection of recombinants, non-transformed, cannot grow on ampicillin or tetracycline medium. If after insertion of a gene into a vector, we will transfer that vector into a host cell. Then we will select. What will happen means, we will get non-transformed cells, transformed cells with non-recombinant vector and transformed cells with recombinant vector. On these three, we want only transformed cells with recombinant vector. That means the cells in which the vector containing our gene of interest. First one is non-transformed cells means that medium will contain both ampicillin and tetracycline. If non-transformed cells means that will not grow on that medium, because that cells will not be containing any of the resistant marker, like it will not contain ampicillin resistant gene, it will not contain tetracycline resistant gene, because there will not be any transformation occurred in that host cell. First one, so we have to clean off that non-transformed cells. Next one is transformed cells. Only transformed colonies can grow in ampicillin or tetracycline containing medium. If a cell is transformed, but we do not know that our gene of interest containing vector has transformed, that we don't know, just transformed. It can grow, because if it is transformed means it will contain the selectable marker. Next one is transformed. Next one, ma'am. Transformed with recombinant vector. This is what we need, because what we need is our gene of interest containing vector containing host cell is needed. As I have already said that there occurs a process called insertional inactivation, where we have inserted our gene of interest into the tetracycline resistant gene. Then after the insertion process, what will happen? That tetracycline resistant capability will be gone and it will be sensitive to that tetracycline antibiotic. Based on that only we are going to select. Ampicillin or tetracycline containing medium. Transformed with non-recombinant vector will be growing. Transformed with recombinant vector will be growing only in ampicillin containing medium. It will not grow on tetracycline containing medium. Based on that we can select the cells which are not growing in tetracycline are said to be transformed with recombinant vector. Next ma'am. Based on replica plating only we can easily pick up the colonies containing recombinant vector. This one is ampicillin. Here this orange colonies are nothing but the cells containing plasmids with R gene of interest, that is transformed colonies with recombinant plasmid. This orange one, that colonies are nothing but just transformed colonies. Then we have to use replica plating method. From that replica, from master plate, then we have to transfer to the normal plate. From that master plate we have to select which colonies are not growing in tetracycline medium. From that comparison we can easily pick up the orange colonies and we can finally grow in our medium. Then finally we will go for expression process and we will get the products. Thank you so much.